Hi, in this video you will learn how to virtually put the sample on chip within the Cell Explorer software. In order to tell the software which sample you have loaded, open the chip and click on Add Sample. There you have to select the right sample type, either tissue section or cell solution. So click on Cell Solution and on Generate New. Here you see 3 pink arrows for sample source, sample prep and sample on chip. In this part of the electronic lab book, you define which kind of sample you have put onto the chip. You record if this was a human being sample or a sample from a mouse or from another species. You also record if there were any in vitro modifications after drawing the sample. And finally, you define how you actually put the sample onto the chip. Now let's start with step 1, the sample source. Click on the sample source arrow and select the organism. In this case, it's a human being. In case you haven't put previously any samples from this particular human being on chips, you have to create a new sample. So click on Generate New and enter all the necessary information about this particular person. Within this form, you don't need to fill out all the fields. Only fill out those that you are interested in being recorded. Let's start with the age. For example, let's enter 30 years and click on Go. I'm not going to enter the weight, but I am going to record the gender, for example, female. For the genetic background, I'm also not going to enter anything. The patient ID, if this is a patient from a clinical study. The health status, for example, if it's a diseased individual or a healthy individual, healthy control. And you can also enter the particular disease that this patient has. And also you can enter a file path or a URL to the health record if you want to. Now select the owner of this object. If you click on Add, you'll get a list, and I'll select Test User 3 in this case. So the owner is a person that has been previously entered into the system. You can also select several owners. And click on Go. Now we successfully completed step 1, entering the sample source. Step 2 is to record the procedure of sample preparation. Click on the sample prep arrow and select the date when you started to prepare the sample. Select the duration of sample preparation, in this case it's 1 hour, and the person who prepared the sample. You can either scan the ID of this person or enter the ID or select the person from the database. In this case, I'm going just to enter the ID. Now select in which solution you diluted your sample. Again, you can either enter the ID of the solution or scan it or select it from the database. With the time, you can get a lot of solutions put into your database. In order to be able to find your solution quicker, you can use the Select Subcategory menu. In this case, it's a buffer, and it's a non-permeabilizing buffer. So there are still a lot of non-permeabilizing buffers in the database. Thus, I'll start to search for the item name. Type in wash and you'll find the ZKWApp wash buffer. Click on it and click on to select and go. Now press the tab button on your keyboard, click go and click next. You have generated a new sample and you can select if you would like to print out the barcode for the sample. In this case, I'm not going to print the barcode, so I select none and click go. The last step now is to describe how you put your sample on chip. Select the sample on chip arrow, click on it, and tell the computer if the cells or your tissue sections have been fixed after they were immobilized onto the chip. In this case, I select yes. Now record when you started to put the sample onto the chip. Please select who put the sample onto the chip. Again, you can either scan or type in the barcode, or you can select it from the database. The cell solution and the microfluidic channel IDs are already put in here, so you just have to confirm if it's the right one. Just click Tab or press Tab, Tab again, click OK or Go. And now you have to select the fixation solution, because previously I selected that I did fix the cells onto the chip. I now have to tell the computer with which solution I actually fixed them. Here again you can either scan the ID of the fixation solution, or enter it, or select it from the database by clicking on this blue arrow. I'm going to select it from the database. In order to find the right fixation solution within the database, I'm going to use the search via the item name. Entering fixation already finds the ZKWApp fixation buffer. So I click on the ZKWApp fixation buffer, click on Select, Go, and Next. At this point, please select the first scan that you're going to do on the chip. So select the number of positions you're going to scan. For example, 10 in this case. Now, the first scan is usually a bleach scan followed by a background scan. So we do the bleach scan with the All filter, click on All, and automatically 20 seconds are the default setting for the bleaching. But you can also change the 20 seconds to another bleach time, for example, if your sample contains very autofluorescent cells. So you click on 20 seconds, change bleach time, select yes, and select, for example, 30 seconds or 10 seconds or whatever you would like to select. Click onto OK. For selecting or for recording the background, please select the channels that you're interested in. Now click to generate scans and exit. Now on the left side of the screen you see the new sample. If you click on Show More, you can see all the details that you previously entered about the sample. You can also change the sample later on by clicking on Change Sample, Yes, and changing any of the steps that you would actually like to change. Now imagine you have drawn blood from a patient, you have processed the sample and have put the same sample onto multiple chips. You don't need to enter all the information about this particular sample again and again. You only need to do this once. In this example, let's check the ID of the sample that we just recorded. Click on Change Sample, Yes, and write down the sample ID. The sample ID contains all information about sample source and sample preparation. After having written down the sample ID, click Exit. Search in the database for the second chip where you put the same sample. Open it. Click again on Add Sample. Select Cell Solution. And this time you don't want to generate a new cell solution, but you rather want to select an existing solution from database. Now let's look in the database for this sample by typing the ID into the LIMS ID field. Click on the sample, click Select, and you see that 2 out of 3 steps of putting the sample onto the chip are now green. You only have to define the last step where you say if the cells or tissue sections have been fixed after being put onto the chip. And again, you fill in the fields start, executor, and fixation solution. You again select which scans you are going to have the first time, how many positions you want to scan. And that's all. You're done.